Alright, so here we are with gluconeogenesis, another component of carbohydrate metabolism. In this case, we're taking something simple and making something more complicated, so this would be an example of anabolism. A-N-A-B-O-L-I-S-M. Okay, so our major pathways in carbohydrate metabolism, at this point in time, we have already talked about glycolysis, and that's glucose to pyruvate. And then we also talked about the fates of pyruvate, one of which was lactate, another was acetyl-CoA, and then, acetyl-CoA for the TCA cycle. But then we talked about homolactic fermentation as well as alcoholic fermentation. Now, another fate of pyruvate, which isn't really a fate of pyruvate, it kind of is, but anyway, another direction that pyruvate can go in is it can be metabolized back up to glucose, which is where we talk about gluconeogenesis. So, the formation of gluconeogenesis, that is, the formation of new glucose molecules is gluconeogenesis, hence gluconeogenesis, new production of glucose molecules. So this is coming from non-sugar precursors, lactate, pyruvate, and glycerol. None of these are sugars. Another example of a molecule that can be used to make glucose is there are some alpha amino acids that can do it. Gluconeogenesis is primarily going to occur in the liver, and under starvation conditions, however, the kidney can make small amounts of new glucose, but largely it's going to be taking place in the liver. Now, between meals, your blood glucose levels are maintained by the hydrolysis of liver glycogen. So glycogen, that long chain and branched chain of glucose molecules, those can be hydrolyzed into individual glucose molecules so that they can be metabolized so your blood sugar doesn't plummet necessarily. Okay, when liver glycogen is ultimately depleted during prolonged fasting and vigorous exercise, that's when gluconeogenesis as a pathway is turned on so that the body can be provided with glucose. I don't know why that, anyway. So you can take something like pyruvate or lactate or glycerol or an amino acid and build it up into glucose. Now this is important because the brain and red blood cells are exclusively dependent on glucose for energy. Overall, when blood sugar levels are low and liver glycogen is depleted, 7 of the 10 reactions of glycolysis are reversed, which, if you think about when we talked about glycolysis, 7 of our 10 reactions were reversible. But under these conditions, when blood sugar levels are low, that's when they're actually going to be reversed. So think about it as they're capable of going in either direction, but only under certain conditions are they going to be going in a particular direction. So when blood sugar is low, well then they're going to be going upstream, whereas when blood sugar is high, you're going to break it down, or you're going to be going in the opposite direction. The 3 steps of glycolysis/gluconeogenesis that are irreversible are 1. Hexokinase, our very first step, 2. PEPCK1 or phosphofructokinase 1, which is the first committed step of glycolysis, and last, pyruvate kinase. So these 3 steps right here, hexokinase, pyruvate kinase, and phosphofructokinase, are irreversible in the context of glycolysis. So when you get a glucose molecule and it gets phosphorylated, well, it can't be dephosphorylated by hexokinase. That is an irreversible step. PEPCK1 is going to phosphorylate glucose 6-phosphate or fructose 6-phosphate to make fructose 1,6 bisphosphate. Well, that cannot be undone by phosphofructokinase 1. And finally, pyruvate kinase, where you are removing a phosphate group from your phosphoenol pyruvate molecule, well, that cannot be simply undone by pyruvate kinase. Each of these 3 steps needs to be basically bypassed. So in order for gluconeogenesis to take place, these three steps have to be bypassed, and you're going to have to use different enzymes in order to do that. Here are those three steps that are kind of laid out as we spoke a moment ago. Hexokinase, phosphofructose kinase, aka PEPCK1, and pyruvate kinase. Those are our three steps. Now, our gluconeogenesis reactions are essentially going to be the same, but a little bit different. Gluconeogenesis, in a nutshell, is taking two pyruvate molecules, which are 3 carbons each, and converting them into glucose, which is one 6-carbon molecule. Okay, so carbons aren't coming out of nowhere. Instead, you have 2 3-carbon molecules that are combined to make one 6-carbon molecule. So mainly, this is done by reversing those enzymes in glycolysis. Now, the synthesis of glucose is the exact opposite from the breakdown of glucose, in the sense that this is going to require 2 NADH, 4 ATP, 2 GTP. Now, our overview of what gluconeogenesis consists of. Well, we've got to start with pyruvate, but it's not quite as simple as pyruvate goes in this direction and boom, it's done. Instead, pyruvate will go into the mitochondria, be converted to oxaloacetate, subsequently converted into malate. Malate is then able to be transported through a malate shuttle out of the mitochondria. That malate can then be converted to oxaloacetate, which can then be converted to phosphoenol pyruvate, which goes the direction of glucose. Those are the steps that are the opposite steps that of glycolysis. So our first reaction is the conversion from pyruvate to oxaloacetate. Now, that is going to be converted by an enzyme known as pyruvate carboxylase. What this is going to do is it's going to add a carboxy group, or a carboxylic acid, onto our methyl group on pyruvate. So in order to do this, we have to invest ATP. Now think about this. We have 2 pyruvate molecules and we're going to get 2 oxaloacetate molecules, because remember, each pyruvate is 3 carbons. And we need to get ultimately up to 6 carbons for, I'm just going to write glucose. So what we're going to do is we're going to hydrolyze ATP. So the energy involved in the hydrolysis of ATP and subsequent conversion to ADP and PI, this is metabolically irreversible. We're going to add our carboxylic acid onto basically our head of pyruvate to make our oxaloacetate molecule. This reaction, it's very important, takes place in the mitochondria. Okay, so this is our first step in bypassing the steps of glycolysis, or bypassing the irreversible steps of glycolysis. So we can't really just phosphorylate pyruvate and say we're good to go. Instead, what we need to do is first we need to carboxylate it to make oxaloacetate. That oxaloacetate is subsequently going to go through what's known as the malate shuttle. Now, what's a little bit complicated here is that this oxaloacetate, also known as OAA, needs to get outside of the mitochondria, because remember, our carboxylation of pyruvate to make oxaloacetate, that took place in the membrane, in the mitochondria. Now, this oxaloacetate needs to get out of there. So I would say that this is analogous almost to the pentose phosphate pathway, in the sense that we've got these carbons that we need to do something with them, but we can't really do anything with them in their current state. What I mean by that is this oxaloacetate is in the mitochondria. We need to get it out of the mitochondria, and we can't just throw it out. That inner mitochondrial membrane is impermeable to oxaloacetate, so oxaloacetate can't simply float out of the mitochondria. Instead, we have to use what's known as a malate shuttle. Now, what the malate shuttle is going to do is it's going to take our oxaloacetate and convert it to malate. with an enzyme known as malate dehydrogenase. And this is specifically a mitochondrial enzyme. So OAA, oxaloacetate, is converted to malate. Okay, now what's the point of that though? Well, malate has a transport protein that's able to move it across the mitochondrial membrane. So we've got this transport that can take place there. Now, malate, once it is in the cytoplasm, well, we've got the same enzyme, malate dehydrogenase, that we use to convert oxaloacetate to malate. Now we're going to do the exact opposite. So our mitochondria, or our mitochondrial malate dehydrogenase and our cytoplasmic malate dehydrogenase, they do opposite reactions. So mitochondrial is going to take OAA to malate. Cytoplasmic is going to take malate to OAA, because oxaloacetate can then be converted to phosphoenol pyruvate by an enzyme known as phosphoenol pyruvate carboxykinase in the cytoplasm. Okay, so to visualize that, we've got this right here. We had oxaloacetate, so this would be MDH, malate dehydrogenase. Malate could be transported across the membrane here, putting malate into the cytoplasm. We have MDH here that converts malate into oxaloacetate. I'm going to go ahead and scribble this. You do not need to know this reaction out, because I think it's I'd be curious to know what's the difference and what's the similarity between these two reactions. Arguably, the reactions are the exact same, but based on where it's expressed, based on where that protein is found, it's going to do two different things. They're doing the exact same reaction, but just in opposite directions. So it's important to remember that oxaloacetate in the mitochondria is converted to malate. Oxaloacetate is a product in the cytoplasm. So these reactions are very similar to one another. So that oxaloacetate, ultimately produced from malate dehydrogenase, can then be converted to phosphoenol pyruvate. Now, that is basically one of the big steps in getting us back into the throes of what are our reactants and products for gluconeogenesis. Now, the enzyme PEPCK carboxykinase, phosphoenol pyruvate carboxykinase, takes oxaloacetate, which is the molecule we went from pyruvate to OAA to malate to OAA. Now what we're going to do is we are going to use a molecule of GTP, and we are going to remove this carboxyl group. Okay. Now, one thing to keep in mind, though, is we've got two molecules of oxaloacetate, and therefore we're going to invest 2 GTP. We're going to remove 2 CO2 molecules, and we're therefore going to produce 2 phosphoenol pyruvate molecules. So we have all of this taking place. And now if you reflect back on what does gluconeogenesis require, I hope that you're now seeing, well, we've got 2 GTP. Both of those GTP are invested right here. We're adding, our one good question, I think, is where does this phosphate come from? Well, that phosphate comes from GTP, not ATP. So GTP, guanosine triphosphate, is equally a phosphate group contributor as ATP is. Okay, so phosphoenol pyruvate carboxykinase hydrolyzes GTP, transfers a phosphate group onto, and simultaneously removes a carbon dioxide from oxaloacetate to make phosphoenol pyruvate. So this overcomes this is basically the reverse of phosphoenol pyruvate to pyruvate. But notice how it took a couple of different steps. It took, well, going through all of this right here, getting through to make oxaloacetate and get it into the right place. Then we could go on to do phosphoenol pyruvate. So our next big reaction is, and phosphoenol pyruvate all the way up to fructose 1,6-bisphosphate, we have a number of different reactions that took place. Our next step is, or our next major reaction is reaction number 2, the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. This is catalyzed by fructose 1,6-bisphosphatase. So a phosphatase is a type of enzyme that removes a phosphate group from a molecule. So fructose 1,6-bisphosphatase, well, that's removing a phosphate group from fructose 1,6-bisphosphate. This is an irreversible hydrolysis, and what it is doing, it's bypassing phosphofructokinase 1. The next step, and the last step, is undoing basically the work of hexokinase. We've got to remove our phosphate group from glucose 6-phosphate. So we're going to use the enzyme glucose 6-phosphatase. This enzyme is only found in the liver and in the kidneys. This is an irreversible hydrolysis step and requires H2O. All right, so 3 steps catalyzed by a number of different reactions. And it required a number of different players involved, transport outside of mitochondria and so on. So this is, in a nutshell, everything that happened. In order to go from pyruvate back to glucose, one of the first hurdles that we had to pass was we had to get from pyruvate, well, to oxaloacetate, and then from oxaloacetate to phosphoenol pyruvate. Then it was a little bit simpler. We just had a pair of phosphatases. We had to remove phosphate groups from fructose 1,6-bisphosphate and glucose 6-phosphate. Some gluconeogenic precursors. There are several metabolites that can serve as precursors by the liver to form glucose. These metabolites are referred to as glucogenic, so they can be used to make glucose. The 3 most important glucogenic precursors are lactate, glycerol, and alanine. Alanine, that's an amino acid, a very simple amino acid, but nevertheless it's an amino acid. Now, lactate can be released by red blood cells and other cells lacking in mitochondria, or that have low oxygen concentrations. Now, in a cycle known as the Cori cycle, lactate is released by skeletal muscle. Once the lactate reaches the liver, it is converted to pyruvate, or it is reconverted to pyruvate. Now, what's the fate of that? Well, this enzyme known as lactate dehydrogenase does that, and now all of a sudden we've got a molecule that, what's it going to do? It's going to go to OAA, to malate, back to OAA, and then PEPCK, or phosphoenol pyruvate carboxykinase, is going to convert this OAA into pyruvate, or sorry, into phosphoenol pyruvate. So it's more or less make this modification, and then it's doing the exact same thing that we just talked about. Okay, glycerol. Glycerol is obtained from degradation of fats in adipose tissue. So breakdown of a fatty acid or a triglyceride will result in glycerol availability. Now, this is transferred to the liver through the blood. Once it's in the liver, glycerol is converted to dihydroxyacetone phosphate, which we've heard dihydroxyacetone phosphate. Dihydroxyacetone phosphate was one of the products from the enzyme aldolase. So that aldolase is going to produce dihydroxyacetone phosphate, and that's going to enter gluconeogenesis, and now we're back going towards glucose. Now, when exercising, muscle produces large amounts of pyruvate. Some of this is converted to alanine, an amino acid, by the enzyme, or by a transamination reaction with glutamate. Okay, so once alanine then reaches the liver, the same transamination reconverts the alanine back into pyruvate. And what's that pyruvate going to do once it's in the liver and ready to go? Well, I hope that was helpful, and I will see you in the next chapter. It's going to go on to produce, it's going to go back up the chain and make our gluconeogenesis. Now the three most important precursors, just talked about those, lactate, glycerol, alanine. Get into pyruvate, it's doing the exact same thing, going to oxaloacetate, to malate to oxaloacetate, back to phosphoenol pyruvate.